Hello friends and welcome to my YouTube channel Way to Success. In my previous video, I have already discussed about one of the major pathway, that is your receptor protein tyrosine kinase pathway, and we have discussed one major pathway, that is your RAS MAP kinase pathway related to this receptor protein tyrosine kinases, right? Now the second pathway is a PI3 kinase pathway. It is also a kind of your receptor protein tyrosine kinase pathway, right? So PI3 kinase pathway, it plays a major role in your insulin production and glucose transport. We will see how this pathway works, but before that we should be clear about certain things, right, which will prove to be the milestone in the understanding of this particular pathway. In my video on GPCRs, I have discussed certain things, right? I have discussed two cases regarding the blood glucose level, right? That is the glucose level in the circulation. First of all, if the blood glucose level will decrease or falls below a certain level, the alpha cells which are present in the pancreas, right, will secrete a hormone that we called glucagon, right? What it will do then, this glucagon will act through the GPCRs, right? This glucagon will work as a stimulus and will act on the G protein coupled receptors. There it will interact with this receptor and activate this receptor, leading to the stimulation. What will be the stimulation? The stimulation will be the breakdown of your glycogen into the glucose, and ultimately it will lead to the increase in the blood glucose level. Second thing, what can happen? If the blood glucose level will be elevated or increased in your blood circulation, this will lead to the secretion of another kind of hormone that we call the insulin, and this insulin is secreted by the beta cells of pancreas, right? What then happens? This insulin will act as the signal molecule, right? It will act as the extracellular signaling molecule and will interact with the insulin receptors, and these insulin receptors are nothing but the protein tyrosine kinases, and this will ultimately stimulate the uptake of glucose and increases the glycogen and also the triglycerides, right? Though their synthesis will be increased, but what happens? It will lead to the decrease in your gluconeogenesis, that is your generation of glucose, right? What you have to keep in mind, that we have discussed about two things here, alpha cells and beta cells. In CSIR net, they can also ask you questions like this, that what kind of cells generate glucagon, right? And what kind of cells secretes the insulin, or insulin is secreted by the alpha cells or the beta cells, or certain other examples will be given and they will ask you this kind of question. So you should keep in mind these small things which you can forget, right? You should not forget these things, right? What is the cell which is producing your insulin, your glucagon? So you should know, right? And you should keep these points in your mind for future purposes. So in this video, basically we are going to learn about the insulin receptors, how the insulin receptors are working, and how this insulin is getting produced, right? Now moving towards the structure of the insulin receptor, right? You can see the images here, right? Insulin receptor is composed of two things. First one is your alpha chain and your beta chain, right? This alpha chain is present extracellularly, right? It is present extracellularly and contains the insulin binding sites, right? Whereas your beta chains, they has three regions. First one is your extracellular region, your transmembrane region, and your cytoplasmic region, right? So what happens? The alpha and beta chains, they are linked together by the disulfide bond, right? Because of this disulfide bond, these two things are linked together, right? These are linked together and these are linked together. These alpha subunits are linked with the beta chains and they form a dimer with another molecule of your insulin receptor, right? With the help of your disulfide bonds. So the two alpha and beta heterodimers are held together by the disulfide bonds between the alpha chains, right? They are already present in the dimer form, unlike the RTKs we read, right? Those RTKs that dimerizes after the activation by certain signal. But if we see the case of this insulin receptor, these insulin receptors, though they are RTKs, but they are already present in the stable dimer form, even if they are not active, right? So further you can see that in this case a single insulin, this is your insulin molecule, and a single insulin molecule is binding to this insulin receptor dimer, causing it to repositioning. You can see the difference. Repositioning is been occurring in this case, and ultimately what happens? The ligand binding domains lead to the repositioning of this receptor, leading to the tyrosine kinase domains on this side of the cell to come into close proximity. Now these two domains are in close proximity to each other. This results in the transphosphorylation of your receptor, right, leading to its activation. I have already discussed what a trans autophosphorylation is. So if you want to learn what is it, you can watch my previous videos, right? So I have discussed all the points here. You can just jot them down in your notes. So have you all got this point? Now what happens next? After following this activation of the kinase domain, the receptor will get autophosphorylated, that is, it's phosphorylated, your tyrosine residues which are already present in your receptor will get phosphorylated, and this autophosphorylated sites will then recruit certain signal proteins. I have discussed the signal proteins with their domains in my previous video already. You can go there and you can learn that from those videos, right? So what is happening here? These autophosphorylated sites now recruit certain signal molecules, right? And these signal molecules have certain domains like your PTB domain and SH2 domain. With the help of these domains, these signal molecules will interact with this phosphorylated tyrosine residues. Now in this particular pathway, a signal molecule which is used and which is working here is your IRS signal protein, right? IRS means your insulin receptor substrate, which is a docking protein, and this protein binds to these autophosphorylated tyrosine kinase residues, right, with their PTB domain. These IRS have a particular domain, that is your PTB domain, to which they will act on these phosphorylated sites, right? Now we have a structure of this IRS, right? You can see certain points here, right? This one, and this one. So it has a particular structure. It has certain regions and domains present in this. So first of all, it has the N-terminal pH domain. pH domain we have already discussed in it when we were discussing the phosphatidyl ionositol pathway. There I have told you pH domains, right? You can watch that video of mine. So here you have the N-terminal pH domain. With the help of these pH domain, this will interact with the plasma membrane, right? Next is your PTB domain. It has a PTB domain with the help of which it will bind to the tyrosine phosphorylation sites which are present on the receptors. Third, we have a long tail containing the tyrosine phosphorylation sites. These sites provide the docking site for other signaling proteins, and with the help of these sites, right, the another protein, it could be anything, which are having the SH2 domains present in them, that protein can bind to this phosphorylated sites, right? And one of this example is your PI3 kinases, right? We are going to discuss it in detail in our pathway, right? So this was all about IRS, and you can jot down these points from here by pausing the video, right? Now moving forward, I have discussed this pathway in my video related to the phosphatidyl ionositol derived second messenger pathway, right? These we have seen how phosphatidyl ionositol is converted to the PIP first with the help of certain kinases, and then this PIP will be converted to this PIP2 with the help of another kinases. And moving in the steps further, this PIP2 will be ultimately converted to this PIP3 with the help of a kinase that we called PI3 kinases, right? And this PIP3, that is called the PI345 trisphosphate, will remain in the plasma membrane, the cytoplasmic leaflet of your plasma membrane. We will see that how this PIP3 will be working in the insulin production and glucose transport. These PI3 kinases, they have two subunits, right? First subunit is your SH2 domain, and second one is your catalytic domain. Right? These PI3 kinases can get activated by binding to the tyrosine phosphorylation sites which are present on the receptors, right? And with their SH2 domains, they already have this SH2 domains, and with the help of these SH2 domains, they can interact with the tyrosine residues which are already phosphorylated of your receptors, right? And ultimately helps in the phosphorylation of this PIP2 to your PIP3. This PIP3 will remain in the plasma membrane cytoplasmic site. Now this PIP3 which are present at the cytoplasmic site of the plasma membrane, this will provide the binding sites for the pH domain of certain signal molecules. I have already told you, right? Now in this case, the signaling proteins which bind to this PIP3, they are of two types, right? One is your PKB, that is your phosphatidyl inositol kinase B, right? And second one is your PDK1. Sorry, this is not phosphatidyl kinases, these are the protein kinases B. So two kinds of signal molecules or signaling proteins which are getting activated is your PKB, that is your protein kinase B, and PDK1, right? Now this PDK along with one more kinase, that is your mTOR, it will activate this PKB, right? And once activated, this PKB will dissociate from the plasma membrane. It is attached to the plasma membrane, and once it is activated, it will dissociate from this plasma membrane and moves into the cytosol or nucleus, right? This PKB mediates the insulin response. This PKB generally have so many functions. One of them is your insulin response or insulin generation, right? These pathways lead to the translocation of the glucose transporter to plasma membrane also. So one of its function is the insulin production, and second one is your glucose transportation, right? How this transportation works? When the insulin level increases in your cell or outside the cell, what happens? A signal is transmitted through this IRS-PI3K PKB pathway, right? Through this pathway, which triggers the translocation of a cytoplasmic vessel. A cytoplasmic vessel which is present in the cytoplasm. What happens to it? The translocation of this cytoplasmic vessel takes place to the cell periphery, right? This vesicle fuses with the plasma membrane, delivering the transporters to the cell surface. These glucose transporters, these are stored in the walls of this cytoplasmic vesicle, and at the surface, these transporters mediate the glucose uptake, right? And this glucose is converted into glycogen by a certain enzyme, and that enzyme is known as your glycogen synthase, right? So this was the whole pathway. Moreover, this PKB will also lead to the generation of your glucose uptake. One, it's its pathway. Protein synthesis also occurs because of the response generated by this, and glycogen synthesis, as we have discussed right now. So that's all with this pathway. I think you are cleared with the insulin receptor mediated pathway. So in my next video, I'll be explaining you the tyrosine kinase associated receptors. One of its example is your JAK-STAT pathway. So we are going to discuss in detail in my next video. So till then, stay tuned. So if you like my videos, please give me a thumbs up, and please share to this video to your friends, and subscribe to my channel for more videos like this. If you have any queries, you can drop that in the comment box. Till then, stay focused and study hard. Thank you.